Okay, so as we continue on with our section on what we did last chapter with the bacteria, now we move on to, as you can see, viruses. And when it comes to viruses, there's probably four main characteristics we'd like to talk about. One, is it a provirus? Two, is it a retrovirus? And a provirus is one that you would normally associate with that of perhaps influenza A, B, and people tend to get vaccinations to try to prevent that type of disease. And the reason we do that is it starts to perhaps give you a jump start on your immunity. And that, again, that's a provirus. And a cold would also be an example of a provirus. Then when we talk about retroviruses, on the other hand, those are probably a little more severe, and probably the most common one that we would refer to is that of what we call human immunodeficiency virus, or HIV. And those are two main characteristics, and another two we would like to talk about is the cycles that these viruses go through. One is what we call a lytic cycle, and then the other is what we call a lysogenic cycle. The lytic cycle is going to be one where the virus, or genetic material, when it takes over that host cell, it replicates inside the cell and then kills the cell and then moves on with all of the little viruses that it produced. The lysogenic cycle, on the other hand, is just a little bit different, and what that does is the entire cell then divides. Okay, so let's take a look at some of these characteristics of viruses. Okay, as we can see, it's made of a protein coat, and it's just genetic material. That's what a virus is. Very simple substances. Okay, and as we can see, or we know that they cause disease, like that of the flu, again, the cold, and many different types of viruses, like cold sores is an example of another virus, and that perhaps can enter into what we call the lysogenic cycle as well, because it never really leaves the body, it just keeps reoccurring over and over again. And we sometimes use viruses for genetic research, and one of them is actually using the herpes virus to treat cancer. And that was a portion of the video that we had seen earlier in last chapter. Okay, so as we continue forward, remember, viruses are non-living components of genetic material. Okay, so again, they're considered as non-living because they lack the characteristics that we would associate with organisms like that of bacteria. Okay, as you can see, they lack a cytoplasm or different organelles that make a life possible. Okay, so they cannot carry out functions, and that's why they need a host to actually reproduce, and that's what they're referring to in this manner. Okay, they don't grow and they don't divide as cells do, unless they take over the function of that cell. Okay, as you can see, they vary in shape, and in that case, they also will vary in the functions, or let's say repercussions they have on their host. Okay. So here's some of them characteristics we were talking about earlier, and that what we'll eventually see at some point refer to what we call a retrovirus. And again, what that does, okay, as you can see, because of this enzyme known as reverse transcriptase, then, okay, it inserts into the host genome, or the host cell's DNA, and takes over the function of that cell. And again, that is what we're talking about with that of a retrovirus. Okay, let's see here. Where does that put us? I think we'll do one more. Okay. And in this process, this transcriptase virus then serves as a template. Okay, the host cells transcribe the virus DNA and the ribosome translate into new RNA proteins. And that's what we're referring to. HIV is what we consider as a retrovirus. And when we say it takes over the function of that cell, it can remain hidden for many years. Some people may be a carrier of AIDS, okay, which has not happened yet because they have this HIV virus, which is a retrovirus. And again, it's one that can remain with no symptoms for up to possibly more than 10 plus years. Okay, we will continue on with viruses next time.